Each lab within UVA's Biomedical Engineering department is working on their own specific specialized research. The primary investigators will briefly explain here their personal focus.
My lab's specific expertise is building computer models of the processes that happen inside of cells. So inside of a single cell, our body's made up of billions and billions and trillions of cells. And within each one of those cells, you have lots of different molecules, like proteins and metabolites, that are all interacting with each other. You have thousands of them. They all have different structures. They all do different things, have different jobs. So within a cell, we build a computer model of how all of those thousands of things help each other, talk to each other, work with each other. And then with these computer models, we can start to predict, well, if I put the cell in a different environment, or if I have a drug that comes in and it hits one particular part of that network, what happens to everything else? So we in the lab, we have models for pancreatic cancer, for example, and we've made some predictions about different drugs that we think are useful. What's cool is these models are used for these different medical applications, but there's also some work we've done and others in the field where we've used these models not for medical but for other applications. Mm-hmm. So a really good example is biofuel. And so the idea is that you can actually grow these microbes in big tubs, and they can take corn husks or other kind of garbage living material and convert it into fuel, convert it into ethanol or other things. And there have been some models of bugs that can make that conversion more efficient. And there's a few examples where they've actually progressed and are able to make these bacteria make these very valuable commodities and chemicals. We've built a model of this algae, and so the idea would be to use a model to predict how would you engineer the algae to convert sunlight into different fuels more efficiently.
This is a matrix of the genes and reactions within a single cell that Dr. Papin and his researchers have developed. It shows over 1,000 proteins and the interactions.
So my research combines experimental work, so actually out at a bench where we have pipettes and chemicals and that sort of thing, with computational modeling. So we're programming, building models, constructing models on the computer. And so the general area that we're trying to advance is understanding how blood vessels grow and adapt, particularly in different settings of disease. The idea being that if we can understand how they do it in disease, we can understand how to fix them when they're not growing or adapting properly using drugs or stem cells or other therapies. And so, the general, theme of my lab is that if we can partner the experimental bench work with the computational modeling work, we can really get the best of both worlds from sort of the scientific standpoint, but also the math and the analytical and the equations and formulas, kind of bring those together into one place, and they can play off of one another and ultimately assist one another, but also in the end advance our ability to form hypotheses and to test hypotheses and to understand how these systems are working and how we can tweak them for a therapeutic benefit. When we pull these stem cells out of the fat, we actually think that when they were living in the fat, they were associated with the blood vessels in the fat. So it's one way to look at what we're doing is that we're just sort of moving around cells from one vascular bed, aka the fat, into another tissue that needs help with growing blood vessels. So we're injecting them into the eye to help the blood vessels in the retina sort of establish themselves and I guess, trend towards a more normal state as opposed to a pathological state, which is what happens in diabetic. We've also used these cells, stem cells from fat, to try to heal wounds, chronic wounds, or wounds that have trouble healing on their own, to try to accelerate the healing process. The idea, again, being that these cells are very supportive for growing new blood vessels. They secrete things that make blood vessels happy.
The vasculature, or the blood vessel infrastructure of stem cells from fat that Dr. Pierce Kotler and her associates use in their lab for their investigation, are pictured here.
Okay, so my research lab studies how the heart works, and the job of the heart is to contract and pump blood through the body. And in particular, we're looking at heart muscle cells, which are the cells that contract and generate force. And so we're really interested in how the heart figures out how to control how fast the heart beats, how hard the heart beats, whether the heart should grow, whether certain sections of the heart die. And we're trying to use computer models and experiments with microscopes and biosensors in those cells to identify new drugs or medicines that can cure heart failure.
In this diagram, you will see a transcriptional network made and discovered by the Cardiac Systems Biology Lab and Dr. Saucerman.
I'm really interested in muscle. So as you can see here, we have a lot of muscles all around our body. So we have more than 650 skeletal muscles that you can, these pictures give you a really great view of the fact that muscles are everywhere. So maybe you don't realize it, but you use muscles for pretty much every movement and function of your body. So maybe more clear is when you're walking, you use your muscles. When you're running, you use your muscles. You know that because you feel your muscles getting tired. When you're using your arms, when you're typing on the computer. But muscles are even more subtle, like they're inside your mouth, and you use them to speak and swallow. They're skeletal muscles inside your abdomen here that are really needed to breathe, so to make your lungs expand and contract as they bring air in and out, and you have really strong muscles inside your body that make that happen, that contract and relax as you breathe. You have muscles in your eyes to move your eyes around, to know where to look. So they're everywhere, so one of the things that I think is really neat about muscle, if I can get there, is what's inside of it. So they're kind of motors, right? So in a car, a motor is what makes the wheels turn and then you can go. Well, what's the motor inside your muscles that makes them work? And they're really strong if you think about it. Like in walking, they hold up your body. Even when you're running, your leg actually supports 2.5 times your body weight. So that's a lot. The muscles have to be pretty strong, right? So how do they do that? So this is one of the things that fascinates me, that leads us to lots of our research projects. And this is sort of a picture of what happens inside the muscle. So over here is the whole muscle. And then once you look inside of it, you see that it's broken up into lots of subparts. So the things within a whole muscle will be broken up into subparts we call them fascicles. That's these cylinders here. And then if you even look inside a fascicle, you see what they're called muscle fibers. And then if you look inside a muscle fiber, you see these long strands of sets of proteins, and they're called myofibrils. But then you look within a myofibril, you get this, what we call is like the building block of the muscle, which is called a sarcomere. And inside that sarcomere are tiny proteins that have little tiny motors. So each of those motors are, they're about, they create, so let's see, how many of these motors do we have in each muscle? I think on the order of trillions of those. So they're tiny. This thing right here is 1,000th of a millimeter. So tiny. You need a really strong microscope to see this thing. And inside are a whole bunch of those. So you have all of these tiny motors It's almost thinking about putting every car in the world into one place and letting all of them work together, and even more than that, and that's what you have inside of a muscle. So I'm really interested, kind of from a very kind of, you might say, basic point of view, and how that works. Like how does a muscle put together all of those tiny motors to work so well to make us walk and move our eyes and do all these different things? So what's neat about it is all these muscles are have all the same things inside of them, but as we know, all these muscles do very different things. So what allows them to do that? And what they do is they change how these fibers and fascicles and sarcomeres are arranged. So they kind of change the structure, and then that influences what it does or its function. So we talk a lot about this thing called structure-function relationship. So how you arrange these muscle fibers is the structure, and then how they function, so how they produce a movement, is its function. So that relationship is really interesting, and it's also, that relationship is what ends up being disrupted when somebody has any sort of pathology or a disease or problem with their muscles. Their muscles become really weak, so they're not strong enough. So that's, for example, in people that have what we call muscular dystrophies. There's a lot of different muscular dystrophies that mean that especially children end up being very weak. They can't move, they're in a wheelchair, they can't breathe on their own eventually. So that's all problems with the structure of their muscle, and that influences their function, so how their muscles can work. Other times, another disease that we think about is cerebral palsy, where cerebral palsy is actually a trauma to the brain in a newborn infant. But what happens is, as you have that trauma to the brain, it influences the part of the brain that sends signals to muscles to tell muscles how to work when you're walking or moving. And then over time, children end up with what we call these walking abnormalities. They can't walk normally like most typically developing children. So they have problems with the function of their muscles. And then actually the structure ends up having problems also, because their muscles will adapt to their function. So they end up with muscles that are really tight, that are really small, that are weak. And so we think about how can we help therapies for those muscles. For example, a lot of surgeons will actually try to operate on those muscles to make them work better. And so we use engineering principles to help make those surgeries better, figure out which muscles should be operating on, how they should be operated on, and really to make those muscles work better.
This storyboard details the investigations going on in the M3 Blemker lab that show multi-scale quality from whole body to tissues to cells and within the cells. There are also photos here of experimentation and chemistry procedures used in their research. Here, a researcher in Dr. Blemker's lab, Kyle Chadwick, is tracking the movement of stingray fins and using computer modeling to portray each joint.
So the work that I do in my lab has to do with cells, right? So they should know about cells, hopefully. This is what all of our tissues and our bodies are made up of. And one of the important things that cells do is they respond to their environment. And their environment changes depending upon your age, your health, sometimes the temperature, what you've eaten, how you're feeling. And these changes in the environment, so these things that go on outside of the cell, are sensed at their edges, and then that information runs inside the cells by molecules, proteins and things that are inside, and somehow these cells will interpret that information that's happening among all of these molecules and change their behavior. Sometimes they may divide, so one cell would become two cells. Sometimes they may change into another cell type. So from one cell type, say in the brain, to another cell type. Sometimes they may die. And these are all important types of interpretation, and what that's called is signal transduction. And this is important for human health because this type of interpretation and the responses that cells decide to make when their environment changes oftentimes gets messed up in diseases such as cancer, cardiovascular disease, other inflammatory type of diseases. And so what the hope is that if we can get a more fundamental understanding of how that interpretation is happening within the cell, we could find ways to correct it when it is misregulated during disease.
Dr. James shows here how manipulated fluorescence helps researchers follow what's happening within the cells.
And then the two colors, the red and the green, I'll take that away so you can see the red and the green. These are two proteins that we are interested in and that we think may get turned on and off non-uniformly in tumors. And you can see these are all the same cell, the same DNA. It was all from a clone originally, and yet this red protein here is a part of the cytoskeleton. I don't know if high school students learn about intermediate filaments. It's an intermediate filament protein that you can see is turned on very strongly in this little cluster of cells, yet all these neighbors here are a lot lower in expression, even though they have the same genome.
Here is an interaction model showing all the receptors and a detailed input-output signaling going on within a cell that has been developed by the Cell Signaling Bioanalysis Lab and Dr. James.
So, my area of research, at least the mainstay of my research, is focused on the molecules that allow cells to move and muscles to contract. So if you think about, say, contraction of a muscle, it's really easy to think of there being a muscle under there. You see it every time you eat a piece of steak. But if you start dissecting this down to smaller and smaller and smaller levels, what's really moving is not the cells themselves, but structures inside the cells. And within the field, we actually call these molecular motors because these are single molecules that consume chemical energy to produce mechanical motion. Right? So all of this is boiling down to individual molecules. And one pair of these that we study a lot is called actin and myosin. Actin is kind of like a railroad track. And myosin is a little bit like something that might move on that railroad track, only it works by attaching, changing shape like this, releasing, changing shape, sticking, pulling again, releasing, over and over and over. If it walks along the actin kind of moves along the myosin, right? And that is the fundamental basis of, muscle contraction. It's what allows things like immune cells to crawl around inside the body. They're dependent on actin and myosin too. And there are even things that depend on it that we really don't want around. And some of these include parasites like malaria. Now malaria literally burrows its way into cells. It finds a red blood cell it likes and it kind of sticks on it. And it's kind of banana shaped, it sticks like this, and then it shoves its way into the cell. And how does it do it? With molecular motors. Now what my lab does is we look at these molecular motors from the perspective not of whole organs like muscle, not from the scale of single cells, but we actually measure how far they move and how much force they generate, measured one molecule at a time using advanced lasers.